Just before starting my presentation, I have no any financial interest to disclose in my presentations. My main objectives today is to discuss four main theories or hallmarks of aging, to explain the science behind anti-aging therapies, to discuss the recent advances in the field of anti-aging, and to discuss the impact of mental health on aging. So as you all know, the survival of any organism, it is a balance or a struggle between the biochemical damage and the repair that the organism is capable of. Any problem with this balance leads to pathologies. And before entering into our main topic, I just want to point out how we define health. In 1948, the World Health Organization defined health as a state of complete physical, mental, and social well-being, and not only the absence of disease or infirmity. I just wanted to point this out because as you grow older, it's very difficult to be in a state of complete health according to this definition. Just wanted to put a perspective on what we think a complete health is. In the same manner, how do we define aging? Now, aging, it is a time-related deterioration of the physiological functions that is necessary for survival and fertility. You can also say it is a progressive decline in tissue or cellular function, or it is a gradual contraction of homeodynamic space. When I say homeodynamic space over here, it refers to a biological system. Now, this could be a cell, it could be an organ, it could be a tissue, or it could be a system. And its ability to function as it should be is what we refer to as homeodynamic space. And as we grow older, then there is limitation or inhibition of this homeodynamic space. And that's how we define aging. Or you can just say it's a process of getting older. So we human beings have always been fascinated by the phenomena of aging since millennia. Philosophers like Aristotle and Plato, they were the first who used to philosophize aging. They were the first one who characterized aging and disease as two different entities. And this is one of my favorite quote from David Bowie. Aging is an extraordinary process whereby you become the person you always should have been. Now, a few questions that we need to ask ourselves. Should aging be considered a disease? Like biological aging, should we consider this as a disease? And I'm going to discuss this in more detail in the next slides. Is there a biological clock in us that can be reset? How long do we want to live actually? Or even better question is, how do we want to live when we age or when we grow older? So these are some of the questions that we need to ask ourselves. So this is the article that was published in Frontiers in Genetics in 2015. It's a review article. And basically the discussion in this article is that, is it a time to classify the biological aging as a disease? And we all know, in fact, that aging is a natural process, but the concept behind having this discussion in the scientific community is that if we classify biological aging as a disease, then our focus will be on early intervention so that we prevent aging-related illnesses. To give you an example, as we age, we have brain atrophy. And with brain atrophy, there is an increased chance of getting dementia. And as you get older, a lot of our older patients have come with dementia. So why should we wait till then? And why should we not intervene earlier? If you consider biological aging as a disease, then our focus should be on early intervention. Now, this intervention could be dietary, could be chemical, could be genetic, could be anything. But if we consider that, then there's a possibility that we can prevent certain biological aging-related diseases. And also to go further on that, now we can we have shown that we can improve the lifespan of worms by almost tenfold, of flies and mice by doublefold, and of rats and killifish by 30 and 59% respectively. But this is only possible if we have a goal of prolonging lifespan and if we intervene early in the life. So these are a few of the articles or studies that were done in early 2000s. I was not going into detail, but wanted to show you that the studies show that we were able to extend the lifespan of the long-lived mice. Certain compounds like resveratrol was shown to improve the lifespan of short-lived vertebrate. And also by manipulating certain proteins like PI3 kinase, they were able to increase the lifespan of worms as well. Now, the main topic of our discussion today will be the hallmarks of aging. There are 9 main hallmarks of aging, as you can see in this diagram. Genomic instability, telomere attrition or telomere shortening, epigenetic regulations, altered intercellular communication, stem cell exhaustion, cellular senescence, mitochondrial dysfunction, dysregulated nutrient sensing, and loss of proteostasis. So we are not going to discuss all of them today. I'll mainly focus on 4 of the major hallmarks of aging. The first 2, that includes mitochondrial dysfunction and telomere shortening. These theories or the hallmarks of aging have been studied extensively since early 1990s. And we'll discuss these 2. And then the next 2, that is dysregulated nutrient sensing and epigenetic alterations. These are comparatively newer theories or hallmarks of aging, and they carry a lot of promise on anti-aging therapies. And these are the 4 topics that we're going to discuss today. A lot of other hallmarks of aging are a downstream effect of these major hallmarks of aging, and which we are not going to discuss today. The first topic will be the mitochondrial dysfunction, also called as oxidative damage or mitochondrial free radical theory of aging. Actually, this theory was proposed as early as 1950s. And the basic concept behind this theory is that increased production of free radicals by mitochondria, it is a driving force in aging process. Increased production of reactive oxygen species is what causing the aging. There were a lot of studies done in 1990s that were able to show the extension of lifespan in flies and worms by altering the enzymes that are involved in production of the free radicals. And these are the 2 studies among many of them which were published in early 1990s and 2000s. The first one, where they were able to show the extension of lifespan in Drosophila or a fly by overexpressing superoxidase mutase and catalase. Both of these enzymes are involved in decreasing free active oxygen species in mitochondria. Then in the second one, again, they were able to show the increased lifespan in a worm by reducing the amount of superoxide production during aerobic metabolism. So the problems with the theory of oxidative damage is that this theory was never replicated in other mammals. And as more studies got published, the recent studies, they have refuted its role in aging. We have found that longer living mice and longer living rodents, they actually have higher amount of free radical compared to short living mice. And also there is a concept of mitohormesis that I will discuss in detail in next slides. So the basic concept of mitohormesis is that mild increase in reactive oxygen species or mild damage to mitochondria is actually beneficial for the cellular adaptation. And a lot of studies have been published recently which prove this theory as well. So this is a review article that basically discusses the same thing that we discussed earlier. It also showed a bunch of articles or studies that were done since 2000s that showed that there is no major role of the free radicals in aging. And that's why the topic is free radical theory of aging is dead. Obviously, it might play a certain role, but the scientific community doesn't think that reactive oxygen species or the mitochondrial damage is the major cause of aging. And again, this article also states the same thing, published in 2008 in Current Aging Science. Basically, the available data that is concerning the role of free radicals in longevity, it is contradictory and does not prove that this theory is what is the root cause of aging. So as I discussed before, the concept of mitohormesis, which is a completely newer concept. So the basic concept behind this is that minor mitochondrial damage or mild increase in the free reactive oxygen species, it actually triggers a beneficial compensatory responses, and thus causing improved cellular adaptation and improved cellular aging. So mild respiratory deficiencies, it has been shown to increase lifespan, and we think that it is mainly because of the hormetic response, or called mitohormesis. So the basic concept behind this is that whatever doesn't kill a mitochondria makes the cell stronger. So now in the market, we have a bunch of antioxidant supplements. The antioxidant market size, it was valued at almost 4 billion US dollar in 2021, and it is projected to reach 7 billion US dollar by 2030. And as I showed you, there's almost no data supporting this theory of anti-aging for antioxidants, and especially in humans, there's almost no credible data. The next topic or hallmark of aging that we're going to discuss is called telomere attrition, or also called telomere shortening. So telomere, these are repeated DNA sequences that you find at the end of the chromosomes. As you can see, at the end of the chromosome, we have telomere, and these telomeres, they keep on shortening as the cell divides, unless it is maintained by a certain enzyme called telomerase. But the problem is that most of the mammalian somatic tissues lacks telomerase. And the basic theory behind this is that if the cells keep on dividing, the telomere keep on shortening. And eventually, this is what prohibits the cell from dividing anymore and ultimately causing cellular aging. As you can see, as the cell divides, the telomere keeps on shortening, and ultimately there is no telomere, and then the cell stops dividing. A lot of studies done in early 2000s actually showed there is a good association between telomere and cellular aging. And that's where the theory of this telomere attrition for aging comes from. Now, a few problems with this theory of telomere attrition or telomere shortening includes that the recent studies, they have found that mice who express higher telomerase, they are not necessarily associated with longer lifespan. And also, they could not find correlation between telomere length and the lifespan of animal in the recent studies. So even though some of the mice, they have like longer telomere length, but they were not associated with improved lifespan. And that is what caused the confusion. The other problem with this theory is that even if we are able to develop some telomere-based therapies, then there is a risk of tumorigenesis or carcinogenesis, mainly because these therapies are supposed to work by increasing the cellular division. But by the same phenomena, this process target can be used against defense against cancer treatment. So this is one of the our study that was published in 2006 in epidemiology. This is just a longitudinal study where they were able to show no association between the telomere length and survival among the elderly, especially when age was controlled for. And going back to the market again, we have a lot of telomere supplements we can find in the markets. And these are supposed to increase telomerase. These are supposed to support our telomere. And then these are actually extracted from like the roots of certain plants, seeds from certain plants. But again, as I showed you before, not much data behind these products, especially in human beings, although they are supposed to increase telomerase. The third hallmarks of aging that we're going to discuss is called dysregulated nutrient sensing. And this is actually a newer concept and also has a lot of promising therapies that can be done by using this concept of aging. We'll also discuss regarding certain compounds like AMP kinase, proteins called sirtuins, and the molecule of NAD+, which all have role on this pathway. So for this hallmark of aging, we need to know about this diagram and this flow chart, which I'll take a few minutes to discuss on. This pathway is called insulin or insulin-like growth factor pathway. It's also called mTOR pathway. So basically, whenever our growth hormone receptors or IGF-1 receptors are activated, now this could be activated by diet, it could be activated by glucose, insulin. So whenever this is activated, ultimately what it does is it causes the activation of mTOR transcription factor. Now, when we call mTOR transcription factor, we need to know that this is a bad transcription factor. What this does is that it causes increased cellular division, increased cellular metabolism, it causes increased cellular inflammation. So basically, all the bad aspects of cellular aging is done by activation of the mTOR. And as you can see, whenever we activate these receptors, it activates these proteins, ultimately causing activation of mTOR. The other transcription factor that we need to discuss is FOXO transcription factor. Now, is mTOR is a bad transcription factor? Now, FOXO is considered a good transcription factor. Now, this FOXO transcription factor, it is supposed to maintain cellular homeostasis. It is responsible for removing cellular garbage. It causes, it also activates autophagy. Basically, all the good things about cells is what is done by FOXO transcription factor. So these are the two transcription factors we need to know about, which have role in cellular aging. So other than these two transcription factors, you can see there are certain other compounds. The one is AMP kinase, which also causes inhibition of this mTOR transcription factor, thus helps with aging. And there are a few proteins, signaling proteins that are called sirtuins. And these proteins, there have been a lot of studies in these proteins, and they have found that they're very beneficial for cellular aging. They cause activation of the AMP kinase. They also cause activation of the FOXO transcription factor as well. And also they directly and indirectly cause inhibition of the mTOR, thus help with the cellular aging. As you can see in this diagram, there is something behind this red block, which I kept it hidden. As you can see, it affects multiple pathways in this diagram. It causes the inhibition of the IGF-1, which is good. It causes the inhibition of the mTOR directly, which is also very good. It causes activation of these certain compounds, which are supposed to help with cellular aging. Activates AMP kinase, activates sirtuin-1. Can anyone guess what this magic compound could be that helps with aging? Actually, to give you more information on this one, this is the only thing that has been consistently shown to improve the lifespan in experimental animals since early 1990s. So this actually is nothing other than fasting or calorie restriction. So since early 1990s, there have been a lot of studies showing that caloric restriction or fasting improves the lifespan in experimental animals. And as you can see, it has multiple ways how it does that. So again, talking about the same pathway, activation of these growth hormone receptors and IGF receptors, they have been linked with accelerated aging. And genetic manipulations that inhibit or attenuate the signaling intensity at different levels. It has consistently extends the lifespan of worms, flies, and mice. And this activation also leads to increased cellular metabolism and thus increased chance of cellular damage. And as I discussed before, by inhibiting either the transcription factor mTOR or PI3 kinase or any of those enzymes on different levels, we have been able to increase the lifespan in experimental animals. Now, going back to this topic of sirtuins, which are a family of signaling proteins which are involved in metabolic regulations. There are total 7 enzymes that fall under sirtuin group of proteins. And modulation or change in this sirtuin activity is what regulates processes like gene expression, cell metabolism, apoptosis, DNA repair, cell cycle, development, and even neuroprotection. That is why there is a lot of research being done in these proteins. So as we age, we have found that the level of sirtuins decrease. And we have also found that as we upregulate or increase the activity of sirtuins, this is what alleviates the cellular senescence. And among the 7 enzymes, mainly the sirtuin-1, it has shown to delay the replicative senescence of umbilical cord fibroblasts in humans. And sirtuin-3 is found to be the only sirtuin for which we have evidence, at least theoretical evidence that exists that it can influence longevity in humans. So this article that was published in 2011, they were able to show in this article that by giving metformin in young mice, they were able to improve the lifespan of the mice by as much as 14%. And also they were able to delay the first tumor detection by 22 to 25%. When they were able, they give metformin to these mice as early as 3 to 9 months. And the way the metformin is supposed to work is, again, we can go back to the same diagram. Metformin again causes the activation of the AMP kinase and also activation of these proteins called sirtuins. That is how they are supposed to, theoretically supposed to improve the lifespan in these experimental animals. And this is a review article, again published in 2017, basically discusses the same thing that we discussed before. And this article discusses why sirtuins could be a promising target in slowing down the aging process. And this article, which was published in 2009 in Nature, and focuses on a compound called rapamycin. Now, rapamycin is actually a mTOR inhibitor. It's a direct mTOR inhibitor. And in this study, while giving rapamycin to the mice, they were able to see that they were able to increase the lifespan of a female mice by 14% and male mice by 9%, which further proves the theory of distributed nutrient sensing is a major cause of aging and the importance of that pathway. Again, going back to the same diagram, as you can see, the certain activity is activated by exercises, by calorie restriction, fasting, dietary restriction, by certain compounds that we discussed, like metformin. There's another compound called resveratrol, which also causes activation of sirtuins as well. As you can see, whenever the sirtuin gets activated, you activate AMP kinase, you inhibit mTOR, which is a bad transcription factor. You also activate FOXO transcription factor, which is a good transcription factor. A lot of studies, they also found that the mice who live longer, they have higher amount of NAD plus level. And these mice were more energetic when they have higher amount of this NAD plus molecule. And which again works by the same pathway, by activating sirtuin activity, and thus improving the lifespan or the health span. There has been a lot of studies recently in this compound called curcumin. This is actually extracted from turmeric, and this review article just discusses the role of curcumin in modulation of aging. And again, the way this curcumin is supposed to work theoretically is again through the same pathway. As you can see, low doses of curcumin is supposed to activate AMP kinase. Again, goes back to NAD plus, improvement in the level of NAD plus and improvement of sirtuin activity, and thus having a good impact on the cellular aging and organismal lifespan. So what this curcumin also does is that it increases superoxide production. And as I discussed earlier in the theory of oxidative damage or mitohormesis, mild increase in superoxide is actually beneficial for cells. And this is also thought to be how this compound is supposed to work. Now going back to the market, now we have metformin, we have calorie restriction, and we have a lot of turmeric supplements. Again, I showed you the theory behind it, and there is not much data behind humans that it affects aging in any ways. We also have some NMN supplements in market, and then this is supposed to increase the level of NAD, nicotinamide and dinucleotide. As I showed you before, higher amount of NAD is associated with improved aging in certain animals, but obviously there has been no data in human beings. We also have supplements of compounds like resveratrol, which are also supposed to activate sirtuins. Again, not much credible data behind any of these supplements. Now, the fourth theory of aging that we are going to discuss is called epigenetic alteration. This theory is also called informational theory of aging. And this theory or hallmark of aging is comparatively newer theory. And the basic concept of this theory is that epigenome is the root cause of aging. And when we say epigenome, epi is the word that is derived from Greek, which literally means above. So basically this means above the genome or above the DNA. What we used to think before is that DNA is what is responsible for aging. The breaking of the DNA and then impairment in the DNA repair process. So obviously these play a role, but the newer theory is that epigenome is what regulates all of these things. So this epigenome, it consists of a record of the chemical changes to the DNA and histone proteins of an organism. It is a process that determines how the genetic information is packaged and is ultimately read by the cell. We have found that epigenome is what is involved in regulating gene expression, development, tissue differentiation, and suppression of transposable elements. And this epigenome can be dynamically altered by environmental conditions, unlike genome, which remains static. Now, in this diagram, this is a chromosome, and this is the DNA. So DNA has two main parts. As you can see, this part of the DNA, it is free, and it is not looped around. And this part of the DNA is looped around these specific proteins called histone proteins. So whenever the DNA is free like this, then the switch is turned on, and the DNA can transcribe. But whenever it loops around these proteins, then the DNA is inaccessible, or it is turned off, and it is useless, and it does not transcribe. And our body releases certain chemical tags, which causes certain changes in the structure of the DNA. So whenever these specific chemical tags attach to the DNA, then, for example, the position of these loops might be different. Maybe these loops will come over here, and then this DNA will be free to transcribe. Maybe there will be addition of a methyl group over here, or removal of a methyl group, or addition of a acetyl group in the DNA. So these kind of changes, or this information is what we call epigenome. So as I discussed before, the basic epigenetic changes in the DNA includes the DNA methylation, histone modification, chromatin remodeling. So all of these changes, what they do is that they will create a noise in the transcription. They will cause impairment in DNA repair. They will cause chromosomal instability. And ultimately, this is what thought to be the major cause of aging, or the root cause of aging. So the thing to remember is that unlike DNA mutations, epigenetic alterations, they are reversible. For example, restoration of acetylation by using certain enzymes or inhibitors, it has shown to avoid age-associated impairment in mice. And also by inhibiting this histone acetyl transferases, it has shown to extend the longevity of the progeroid mice. So again, going back to the structure of the DNA, in this diagram, what I wanted to show is that both of these structures are the same DNA. The one is supposed to be young DNA or chromatin, and the other one is supposed to be the older one. And as you can see, when the same DNA gets older, the structure gets is very varied. The loops are in different positions. As you can see, there are new CPG islands. You have some hypomethylation and other changes that can occur during aging. And the basic concept behind this theory is that if in the older DNA there is a removal of a methyl group, why can't we add a methyl group? Why can't we change the structure of the DNA to what it used to be, or at least what it resembled to be before? And this is the whole concept behind the theory of informational theory of aging, or considering epigenetic changes is a major hallmark of aging. And epigenetic changes, it has shown to contribute to many hallmarks of aging that include cellular senescence, mitochondrial dysfunction, and other hallmarks of aging as well. So we think that this is the major factor that downstreams all the effect of aging. Now, the important thing about this is that since these epigenetic changes are thought to be reversed, or we can change this, and this is at least theoretically, and there have been a few studies done on this theory, but if we are able to reverse or change these findings, then this opens a plethora of experiments and therapies that can be targeted towards anti-aging treatment in future. And that is why this theory is so exciting, and a lot of research is being done in this concept. So this article, which was published in 2011, what they were able to show in this article or study was that by giving or supplementing this sodium butyrate to our mice in drinking water, they were able to improve the lifespan. And basically what this does, sodium butyrate, is that it is a histone deacetylase inhibitor. And by inhibiting this histone deacetylase, they're able to remove the defective DNA repair, and that is what they suppose that they improve the lifespan of the mice. And this article, loss of epigenetic information is the cause of mammalian aging. This article actually published in Cell. It was published just like 2 months back in January. The main author of this article was Dr. David Sinclair from Harvard. And in this study, what they were able to show is that by inducing some epigenetic changes causing DNA breaks, they were able to accelerate the aging in mice. Uh, and I will discuss four main points from this article. So, double-stranded DNA breaks erode the epigenetic landscape. So, whenever there is a break in the DNA, then that is how the information or the epigenome changes. And this loss of epigenetic information is what accelerates the hallmarks of aging. And as I discussed before, what this theory thinks is that everything else comes from epigenetic changes. And this is the main hallmark of aging. And these changes, they are reversible by epigenetic reprogramming. And by manipulating the epigenome, aging can be driven forward and backward. And this is what they theorized. So this picture, I got it from the Harvard website and also from the same article, the study that I showed just before. Both of these mice are supposed to be identical twins. These are identical twins. But what they did was that they induced some epigenetic changes in the mice on the right side, causing DNA breakage. And as you can see, the mice on the right side, the skin is more wrinkled compared to this one. You can see the hair is more grayer. The scientists, they were also able to assess the internal organs, and they found that the organs were older as well. And basically, this mice, compared to this one, is not only looks older, but also is now biologically older. Although both of their chronological age is 16 months. This is how they proved that by changing the epigenome, they were able to accelerate the aging in experimental animals. Pretty interesting, if you ask me. So finally, before the conclusion, I just wanted to touch base on the impact of mental health. That includes happiness, a positive self-perception, and the psychology on aging. And we all know that a good mental health is the backbone of good aging. And we have a lot of patients, elderly patients who suffer from many mental issues, and that causes spiraling in their health, that causes early aging. But I just wanted to focus on a few studies that were done which prove this thing. So this article study was done in 2000, published in 2018, and this was done in Singapore. And in this longitudinal study, they involved almost 5,000 Singaporeans, and they used this score called happiness score, standardized score. And what they were able to show is that the likelihood of all-cause mortality was lower by 9% for each unit increase in the happiness score that they used. And they concluded that the happiness is associated with a reduced likelihood of all-cause mortality among older people in Asian population. Now, this is another study that was done by Dr. Levy from Yale University. And this study was published in 2002. And in the study, what they did was that they used the sample that consisted of 660 individuals. And they used the sample from this study, Ohio longitudinal study of aging and retirement. And they matched the mortality data from this sample to the National Death Index. And they were able to conduct survival analysis. And what they found was that the individuals with more positive self-perception of aging, they lived up to 7.5 years longer than those with less positive self-perception of aging. And by using this study and multiple other studies that Dr. Levy published herself, she published this book called The Breaking the Age Code. The other study that I wanted to focus on is this one. So this is one of the longitudinal study that is done on human behavior or human happiness. It is called the study of adult development. And this was started in Harvard Medical School. There have been four generations of researchers or authors who are involved in this study, and the current one is Dr. Waldinger, along with others. So there are a lot of findings from this study, but I just wanted to focus on this one point that they mentioned in their study. Close relationships, it helps to delay the mental and physical decline. And they are better predictors of long and happy lives than social class, IQ, or even genes. And by using the summary of the information from this study, Dr. Waldinger actually published this book called The Good Life, The Lessons from the World's Longest Scientific Study of Happiness. Now, before leaving, I want you to remember 6 main points from this presentation. Early intervention, it has shown to delay aging in experimental animals, but we don't have enough data on human beings. We have various anti-aging supplements that I showed you in the presentation in the market, but they don't have any credible data behind them. Dysregulated nutrient sensing and epigenome, these are considered to be future of anti-aging therapies, and I discussed why and why these two theories are so exciting. Calorie restriction and healthy lifestyle that includes exercise, good mental health, they have a positive impact on aging. Certain compounds like metformin, curcumin, the molecule NAD+, resveratrol, these are found to have, or shown to have anti-aging properties in animals. And the positive perception of aging, quality relationships, and happiness, these are some of the things that we have shown to improve survival in multiple longitudinal studies. So this is a painting that was painted by Lucas Cranach in 1546, and the title of the painting was The Fountain of the Youth. Just wanted to show you that since the dawn of human beings, we have always been fascinated by the concept of youth, or remaining youth. Everyone wants to look younger, feel younger, be younger. And this is one of the songs by one of my favorite band, Queen, Who Wants to Live Forever. And this is one of my favorite quotes from Galen, who was a Greek philosopher and also a physician. The best physician is also a philosopher. So these are my references. And thank you for listening to my presentations.